So hello and welcome to APEC inaugural webinar. So this is a personalized T-cell therapy against hepatitis B virus infection and HBV-related hepatocellular carcinoma. My name is Donovan and I'm the Scientific Program Executive for APEC Regional Marketing Team under the Life Science Research Solution Business of Merck KGaA, Darmstadt, Germany. I'll be the moderator for this webinar. Before the start of the webinar, please note that this webinar is being recorded and the link will be shared after the webinar is completed. You may submit any questions at any point using the question tab located below the video player. We will address the questions at the end. Now I'd like to introduce our speaker for today. So Professor Antonio Bertoletti from MD Program in Emerging Infectious Disease, Duke NUS Medical School, Singapore. Professor Bertoletti is an expert in the field of viral hepatitis. He's the Director of Infection and Immunity Program at the Singapore Institute of Clinical Sciences, A*STAR, until 2013 before joining the Emerging Infectious Disease Program at NUS Medical School. In 2015, he co-founded Lion TCR Private Limited with Dr. Lee, a biotech company developing a new immune-based treatment for virus-related cancers and chronic viral infections. At this point, I will hand over to Professor Bertoletti. Professor, please.
Thank you. Thank you very much for the invitation to speak at this webinar. I want to thank Merck to organize this very nice initiative. So I will start with my disclosure that you can see in this slide. I'm acting as a consultant on an advisory board of Gilead and other company, and also I'm as the co-founder of Lion TCR. So let me start the presentation. First, just to point out the extent of the problem that we are facing when we are dealing with hepatitis B viral infection and hepatocellular carcinoma. So for hepatitis B virus infection, it's important to know that even though we have a very effective prophylactic vaccine, still we have around 300 million people infected over the world. And in particular, there are 75% of all the people that are infected are actually present in Asia, the place where I am located. We have therapy for hepatitis B, effective therapy that, however, are blocking only the replication of the virus, which means that the virus can be inhibited but cannot be eradicated from the patient. And this is why the patients have to continue to take the drugs. And this represents, at the end of the day, a problem, because the reality is that we are not really able to get rid of the virus. The other problem is, again, hepatocellular carcinoma. And here I put in some numbers. There are approximately 600,000 cases of at year, new cases, and about 55% of these new cases of hepatocellular carcinoma, these cases are in China. And what is important to understand is that about 80% of all the new cases of hepatocellular carcinoma are actually HBV related. The therapy of hepatocellular carcinoma is good if you are able to detect the tumor at very early stage. In particular, if you have a liver transplantation or the resection, you can have actually a survival that is about, in 5 years, is about 70%. The problem is that, however, the tumor is usually completely asymptomatic, and therefore it's very difficult to get early diagnosis. And the problem therefore starts that the survival time for the advanced hepatocellular carcinoma with the present therapy is extremely poor. And you can see here, as I've written in this slide, that is about only, let's say, 15 months of survival with sort of double therapy with TACE and sorafenib. Okay, now let me go to the focus of this talk, that is about our attempts to produce and to develop this personalized T-cell therapy in HBV-related hepatocellular carcinoma and in HBV infection. And what we are doing is basically to engineer lymphocytes by introducing specific T-cell receptors that are specific for HBV epitopes that are presented by HLA. So basically making new T-cells that will be able to recognize both HBV-infected hepatocytes or hepatocellular carcinoma that can present HBV epitopes because the HBV can integrate in hepatocellular carcinoma. So let me tell you, therefore, how I'm going to organize this talk. I will discuss first why we engineer HBV-specific TCR-directed T-cells. I will explain briefly how we do it. And then I will show some data on what these cells are able to do both in animal model and in patients. Okay, let me start with why do we want to engineer HBV-specific TCR therapy for antiviral therapy of chronic hepatitis B. And I'm starting with these slides that basically say that the patients with chronic hepatitis B do not really have a good adaptive immune response. Basically, their T cells and their antibody are present at very, very low frequency in patients with chronic hepatitis B. But at the same time, we know that patients that are instead able to resolve hepatitis B viral infection, they have T cells and antibody able to recognize the virus. Therefore, we know from the data that have been done by several groups that HBV-specific T cells are actually essential for the control. But how can we resolve the defects of anti-HBV specific immunity that are present in chronic HBV infection? So there have been different strategies, and I'm just putting in this slide different strategy on clinical, as I say, therapeutic strategies that are trying to boost and to restore HBV-specific adaptive immunity. One is to act with anti-PD-1 and antibody that can be able actually to restore the function of the T cells that are exhausted during chronic HBV infection. Another possibility is to use antibody therapy, and another one is to actually use vaccine, but in this case, therapeutic vaccine, to boost the adaptive immunity that is defective in patients with chronic hepatitis B. Now, without going now in the details of talking about antibody therapy, I will tell you what are the problem of try to restore HBV-specific adaptive immunity using vaccine or checkpoint inhibitor. The problem is that both of these strategies require the presence of some HBV-specific T-cell response. And the situation of the T-cell response in patients with chronic hepatitis B is shown in this particular slide that represent here on the left corner of the slide. You can see here the frequency of T-cells specific for hepatitis B in patients that are characterized with high quantity of HBV replication, basically the patients that we would like to treat. And you can see that the frequency of these T cells is extremely low, actually pretty much almost impossible to be detected. The same is happening in patients with lower viral replication where there is a little bit more cells, but usually these are not exactly the target of our clinic. So the problem of patients with chronic hepatitis B with large quantity of viral replication is that in reality, there are very few T cells that we can try to boost with our therapy, with checkpoint inhibitor and with vaccine therapy. This is why basically we are thinking that these two strategies could not work, and maybe to restore the defective T cell response that is present in chronic hepatitis B, we can actually have to engineer new HBV-specific T cells. Now, let me go instead to explain why we are thinking that also our personalized therapy based on TCR-directed T-cell might also be used in anti-tumor therapy, so against HBV-related hepatocellular carcinoma. And here, the rationale is expressed in this slide. So what we need to know is that in hepatocellular carcinoma, there is about, let's say, 80–90% of hepatocellular carcinoma have HBV DNA integration. This HBV integration, we know, is able to produce messenger RNA. But we also know that in patients with chronic hepatitis B and hepatocellular carcinoma, there are actually no HBV-specific cells. Now, the point here is that we can potentially use TCR-directed T-cells specific for hepatitis B to recognize not only the HBV antigen that is expressed on the hepatocyte, but we can actually use these TCR-directed T-cells to recognize hepatocellular carcinoma that has HBV DNA integration and that can express HBV antigen in a way that is recognizable by our T-cell receptor. And I put here on the bottom of the slides the reference of the paper that we just published in Gastroenterology that is going to explain in more detail what I just told you. But basically, we can actually use the expression profile of HBV DNA integration present in hepatocellular carcinoma to really select the T cells that can be able to be used for this therapy. And if you are more interested, you can go directly and read this paper. Okay, now, one question that usually most of the people are asking, why do you use TCR, so classical T-cell receptor, and not use CAR T-cells? CAR T-cells have been shown to be extremely useful in some other form of tumor, particularly B-cell lymphoma. And, the CAR, that is an abbreviation for the chimeric anti-cell receptor, has some advantages in comparison to classical T-cell receptor. The advantage is that since this chimeric anti-cell receptor is made basically by the constant region of a TCR, but the variable region of a TCR is an antibody, this antibody can recognize conformational antigen present on the surface of the tumors. The advantage here is that the CAR is non-HLA restricted, meaning that we can use the same CAR T cells in many different patients. And therefore, we can use a single product in many different subjects. The T-cell receptor instead is a little bit more complicated because the T-cell receptor does not recognize conformational antigen that is present on the surface of the cells, but recognize epitopes that are presented by HLA class I molecules that are clearly different in different patients, and that they are presenting this antigen that is the product of the endogenously processed tumor antigen or viral antigen that is present within the infected cells. So now it is clear in a way that using a single product in many different patients might be easy, but there are other problems here. The problem is that most of the tumor antigen, like also the viral antigen, are actually endogenously produced and are not present on the surface of the cells. The other big problem in hepatitis B and the problem of using the chimeric antigen receptor in hepatitis B is shown in this slide. And basically is the fact that we need to know that during chronic HBV infection, and both in chronic infected people and in patients with hepatocellular carcinoma, we have large quantity of soluble antigen that is present in the circulation of the patient. And this soluble antigen can therefore block the CAR T-cell function. I try to, let's say, summarize this concept in this slide. This is just an idea that if we have a CAR TCR, so a T cells that have been engineered with a CAR, and the CAR is specific for the S antigen that can be expressed on the surface of hepatocellular carcinoma, but the point is that we have a large quantity of soluble antigen in the circulation of the patient. And this soluble antigen can actually block the CAR T cells. This is not going to happen with TCR because, again, the TCR does not recognize the conformational antigen, but recognize the complex HBV peptide HLA complex on the surface of the cells. And therefore, this TCR will not be inhibited by the CAR, as you can see schematically represented in this slide. So, again, now try to summarize this first part of the talk. Why do we engineer HBV-specific TCR-directed T cells? One is to restore the defective HBV-specific T cell present in chronic hepatitis B, and therefore trying to use our TCR-directed T cell as an antiviral. The other point is that we want to use it, the TCR-directed T cells, in anti-hepatocellular carcinoma therapy, because we want to target HBV antigen that can be produced in HBV-related hepatocellular carcinoma by the HBV DNA integration. Okay, now let's start to discuss briefly how we make our HBV-specific TCR-T-cell. How do we make it? How do we got the TCR? And here is that we are basically we have produced in the past and we are still producing a large library of new T-cell receptors because we have extensively characterized the HBV-specific T-cells in patients who have resolved HBV infection. So, as I told you before, in patients that are able to control the infection, these patients are able to mount a T-cell response specific for hepatitis B. So we therefore are able to select CD8 T-cells from resolved patients. We clone the DNA of the TCR from these patients. And at this point, we have a library that we can put basically the DNA of the T-cell receptor with different methods, and I will talk about it later, in the T-cells of patients. And therefore, again, our our product is basically a library of T-cell receptor restricted by different HLA and, I will say, able to recognize different epitopes that is deriving from CD8 T-cells that are present in patients with resolved hepatitis. And at the moment, in our laboratory and also online TCR, we have basically having a TCR library that can cover approximately 60% of the Asian subject. Sorry, it's written object, but there is a mistake. Okay, let me show you here one data. Basically, what we are doing is to, again, introduce in the lymphocytes of the patients, of the normal individual, depending what we want to do, the information of the T-cell receptor for specific for hepatitis B. And here, on the right side of the slide, you can see some of the results. For example, in this dot plot, you can see the profile of T-cells of patients with chronic hepatitis B that have been stained with CD8 and a pentamer, that is a reagent that is able to recognize a particular T-cell receptor. In this case, a T-cell receptor that is specific for the region core 1827 of the hepatitis B. So you can see here that in patients, in the CD8 T-cells of the patients before transfection, there are no T-cells that are expressing the TCR. But after transfection, we are able to produce large quantities, about 20–30% of the CD8 T-cells that are able to express the known T-cell receptor. Now, what are, let's say, the function? What our HBV-specific TCR-redirected T-cell can do? So, let me show you first some in vitro and in vivo data. The first one is shown here. What basically we have done an extensive characterization of the function of the T cells that have been modified by introducing the TCR, and we can see that these TCR-redirected T cells are basically able to recognize targets that have been passed with peptide or FG2 cells that are infected with hepatitis B. Basically, we are able to make TCR-redirected T cells that are able in vitro to recognize HBV-infected hepatocytes. The important point is here that showing basically what we call sensitivity of T cell activation, where we are going to compare basically the ability of our TCR-redirected T cells that have been produced using lymphocytes of patients with chronic hepatitis B. And we can show that these TCR-redirected T cells have the same sensitivity, so the same basically ability to recognize HBV epitope, like the CD8 clones that we got from patients with resolved hepatitis. What does it mean? It basically means that we are able to engineer T cells that have the same function of the T cells that are present in patients with resolved hepatitis. And here again is better shown this, that we have able to produce T cells that are identical affinity of the T cells that are present in patients with resolved hepatitis, that are these cells that are shown with these black dots. And if we are introducing TCR in T cells that are coming from patients that have low or high level of viral replication, we can actually make T cells that are again the same functionality than the T cells that are present in patients with resolved hepatitis. So we have used these cells in different animal models. In this animal model here, I'm showing the ability of TCR-directed T-cell actually to lyse and control an hepatocellular carcinoma that is expressing S antigen. This is, again, a mouse model. This is a SCID mouse, so a mouse that does not have immune system, where we introduce, we transfer a tumor cell that is a natural hepatocellular carcinoma expressing S antigen. We modify this tumor by introducing luciferase, and you can see that the tumor is basically growing in the sort of flank and a little bit in the peritoneal cavity of the mice. Now, if we are treating these mice with TCR-directed T cells that have been engineered to express a T-cell receptor specific for core, which is not present in the tumor, you will see that the tumor is not eliminated. But if we are treating these mice with TCR-directed T cells, where we introduce the S-specific TCR that is expressed by the tumor, you can see that we can have complete elimination, showing that our TCR-directed T cells are actually able in vivo in an animal model to control tumor that is expressing S antigen. Now, I want briefly to show some of the initial data that we got in men that represent the first proof of concept that we can actually use our TCR-directed T cells in patients. This was a patient that actually had an hepatocellular carcinoma in a chronic hepatitis B. He was transplanted. He stayed well for about 10 years. But after 10 years, unfortunately, he started to develop relapses of hepatocellular carcinoma. We test this metastasis of hepatocellular carcinoma, and we can see that these metastases were expressing the S antigen. So the patients basically have S antigen, so the envelope antigen of hepatitis B that was produced by the tumor, but was not produced by the liver. So we introduce and we treat these patients with TCR-directed T-cell, TCR specific for envelope, and we adoptively transfer very low number of cells. This was an attempt that we have done already about 4 years ago, where we were still not really sure about the potential danger of our T-cells, so we infused very low number of cells. And what we got was that actually these TCR-directed T-cells were able to reduce of about 90% the quantity of S antigen. Again, I want to remind you that this antigen was only and exclusively produced by the metastasis, showing that there was some effect. I want to stress that we didn't save the patients. The patients, when was treated, actually have very large metastases everywhere. And due to, let's say, technical issue, we can start to treat the patients only when it was so advanced. But I want to show these slides just to show how, in a way, our therapy is potentially very efficacious. Because these slides show in the left part what was done to the patients before our immunotherapy. And basically, the patient has surgery and radiation therapy to try to eradicate this metastasis. And you can see here that in the green line is represented the quantity of S antigen that the patient has. He got a surgical removal of the metastasis, and you can see that the S antigen is decreasing. And he has also radiation therapy here. But I want you to show, to make your, let's say, to put your attention to the time. Here is January to September, 9 months. He had this drop of S antigen after surgical removal and radiation. Then the S antigen started to grow again. It was again surgical removal, but nothing happens. This is the level of S antigen reduction that we had in a single month with only 10,000 cells per kilo. And you can see actually that the efficacy was extremely high. Again, I want to stress that these patients survive the therapy and stay well for about 3-4 months. Unfortunately, after 4 months, he died. this again, I want to stress, it was the first attempt with very few number of cells. Okay, now the first conclusion of let's say, this part of the talk is that actually I show you that we can basically engineer large quantity of functional high avidity HBV specific T cells. We show in animal model that these cells are functional in vitro, and we show that they are able actually to recognize S antigen positive hepatocellular carcinoma with HBV integration, also in patients. A sort of first proof of concept that we can use this therapy in patients with HBV-related hepatocellular carcinoma. Now, I want also to somehow discuss with you the problem of this therapy, because it's clear that there could be a problem. And one of the main problems is that actually our TCR-redirected T cells can recognize equally well the tumor and the antigen that is expressed on the hepatocyte. And this means that if we want to treat this, we want to use these T cells in patients that have primary hepatocellular carcinoma in a chronically infected liver, we can have actually the possibility that our T cells will not only lyse the tumor, but will lyse all the infected hepatocytes. And this is a problem. This is a problem because we can have fulminant hepatitis. So how can we basically overcome this problem? We use now two different strategies. The first one is basically now to treat, as I showed you before, only patients that have been liver transplanted and they have XTC relapses. Why we do that? Because the liver transplantation usually is HLA mismatch. And for this reason, in a patient with chronic hepatitis B, where you have a new liver, the liver has an HLA that is different from the metastasis that is originated from the previous tumor. And therefore, we can basically select the T-cell receptor that will recognize HBV epitopes only in association with the HLA that are present in the metastasis and not with the HLA that is present in the transplanted liver. And therefore, avoid the potential destruction of HBV-infected hepatocytes that can occur in the transplanted patients. The other strategy that we're using, and shown on the right side of the slide, is that actually we try to reduce the lifespan of HBV-specific TCR-redirected T cells. And we do that by engineering T cells not using viral vector, but by using direct messenger RNA electroporation. Let me show you what is the point here. The point here is that when we are introducing the TCR with a retroviral vector, we have stable transfection, and it's shown in this figure. This is the expression of a TCR. When we are introducing the TCR with a viral vector, we have the TCR is expressed constantly over time. With messenger RNA electroporation instead, well, first, we don't do any gene modification, and we have only a messenger RNA that can express the TCR only for a short time. And you can see here that the expression is very high at the beginning, about 70, even 80% of the T cells express the introduced TCR, but the expression of the TCR fall down, and at about 5-6 days, it's basically undetectable. So the point here is that this could be a disadvantage because it means that we have to introduce these cells several times, but it's an advantage at the moment because we can really start to analyze in patients how many cells we can introduce without causing any liver damage, because we know already that we have a sort of time-controlled TCR expression that allow us to do multiple doses, different quantity, and understand whether the TCR-redirected T cells are going to be effective only against the tumor or can have some toxic effect. And again, I want to show you in this slide that the electroporated cells that are indicated here as a blue line are, I would say, still effective against tumor, even though that's less effective than the retroviral TCR. So the T cells that are expressing the TCR stably. Here again, it's an animal model that I showed you before. There is this animal that is been developing this hepatocellular carcinoma as antigen positive. As I showed you before here, when we are introducing TCR that have been engineered with the retroviral vector and therefore are expressing the TCR stably, we have a complete elimination of the tumor. And you can see it here in the photo and by the red line on the graph. Where we are introducing messenger RNA electroporated T cells, we can actually see that the tumor is somehow blocked the growth. We are not eliminating, but we are just suppressing its expansion, which again is something that at the moment we can actually use also in patients. So what I want to say is that yes, the messenger RNA electroporated are probably less potent, but at the moment are actually can be used in patients without the risk of toxicity. Now, briefly, I want also to mention that this messenger RNA HBV TCR can have a good antiviral effect, and we show this in an animal model, that is a different animal model. We use a model of humanized chimeric mouse, it's called. It's a mouse where the liver has been reconstituted with human hepatocytes. And this work has been done in collaboration with the University of Hamburg, and in particular with Maura Dandri and Janine Kemp. Basically, these mice can be infected by hepatitis B virus. I have to remind you that hepatitis B virus infects only and exclusively human hepatocytes. Therefore, HBV cannot be used in normal mice because HBV does not infect mouse hepatocytes. So in this mouse that have been reconstituted with human hepatocytes, we can have actually a chronic infection of this human hepatocyte. And when we are introducing our messenger RNA TCR-redirected T cells, so T cells that are going to express the TCR only transiently, we can have a very good reduction of viral replication. And what is important here is that what we want to show is since this messenger RNA have a transient expression of the TCR, we have also transient inflammatory events that are showing in this slide. You can see that when we are introducing our TCR-redirected T cells, we have a peak of transaminases that then resides, which means that again, it can be possible to use these TCR-redirected T cells that are expressing the TCR transiently or also for antiviral therapy. Now, I don't want to go into the details. I want just to say that our laboratory is also working on the antiviral part of, try to expand the potential of using TCR therapy in antiviral by trying to engineer what we call non-lethal T cells. So basically try to engineer T cells that can be able only to inhibit viral replication without causing any lysis of the hepatocyte. This is something that the laboratory is working, again, with the idea to try to use TCR-redirected T cells also in the therapy of chronic hepatitis B. I want to finish my talk just to showing few other cases that we have recently treated here in Singapore, and also some patients in China, in Guangzhou, where again we are treating metastases of hepatocellular carcinoma that are occurring in patients with liver transplant. And I'm going to show in particular the results that we have from one patient that we have studied extensively here in Singapore that have developed metastases of hepatocellular carcinoma after liver transplantation. The story of the patient is pretty much shown here in this insert A, where you can see the patients have hepatocellular carcinoma. He was liver transplanted. He developed multiple metastases in both lung about 7 months after liver transplantation. At that point, the patient starts sorafenib, that is a multikinase inhibitor that is used for the therapy of hepatocellular carcinoma. But nothing happens, and then we start TCR-T-cell therapy at about, let's say, more than 2 years ago. What we were able to demonstrate in these patients, and is shown in this figure, is that actually the retroperitoneal metastasis and the primary hepatocellular carcinoma of the patient were expressing HBV DNA, HBV epitopes. We were able basically to take the biopsy and demonstrate that there was messenger RNA of HBV antigens that was particularly producing an epitope that was presented by a TCR that was restricted by HLA-B58. Now, basically, we were able to understand which was the epitope that was presented in the primary HCC and in the metastasis of the patient, select our TTR, and then use our TCR-redirected T-cells for the therapy of the patients. This slide that is very complicated, and I'm sorry about that, it just shows how many different infusion of the TCR we have done in the patients. Usually at the moment we are starting a strategy where we do this sort of what we call dose escalation, basically introducing and adoptively transfer different number of TCR-redirected T cells. We go from 10,000 cells per kilo to 5 million cells per kilo in about 1 month. We observe the patients, we see, and we want to be sure that we don't have any toxicity of the liver transplant, and then we start a treatment that is basically adoptively transfer 5 million cells per kilo at a sort of 10 days time. Now, we have been treated now these patients for more than 2 years, and I want to show you some of the data that we have. The most important data were actually the data that I'm showing here, and on the left side of the slide, you can see actually the lines represent all the infusion of the different TCR-redirected T cells. The red line represents the volume of the different metastases. Now, I want you to really, for example, focus on the metastasis that is present on the top of the slide, showing basically that after infusion of the TCR-redirected T cells, we have an enlargement of the metastasis, what probably in other anti-tumor therapy you could call pseudo-progression. But then we have actually that the tumor almost disappears. This happened actually in almost every single lesion of the lung, except one that is shown here on the bottom of the slide. So we can actually show that we have some effect in most of the lung metastasis of the patients. This is what I'm going basically now to end my talk. That just with these two conclusions, I think I show you that we are able to engineer HBV-specific TCR-redirected T cells with different methods, stable and transient expression. I think I show you that we can potentially use these cells for both antiviral and anti-tumor therapy. And I start to show you that the HBV TCR-redirected T cells can have some efficacy in patients with HBV-related hepatocellular carcinoma. And I remind you to the paper that we just published in Gastroenterology, where you can actually, let's say, look with much more detail the things that I just told you now. And I want to finish actually just by giving this last slide where there are all my groups and all the many collaborators, because it's clear that such work cannot be done alone. In particular, in addition to thank all my groups that is represented in this slide, in particular Anthony Chan, Nina Lebert, Michelle, Lockman, all the people that are really work a lot on this project. And I want also to thank the Singapore General Hospital, in particular Professor Chow and Dr. Tinesh Krishnamurthy, that have really helped us to introduce our TCR-redirected T-cell therapy in patients. Clearly, I have also to thank many people of the Lion TCR company that have helped us to really bring this new therapy to patients, and many other collaborators. I talk about other collaborators during my talk, in particular Maura Dandri in Hamburg and Dario Campana that have also helped me a lot on establishing this therapy. And I thank you very much for your attention.
Okay, thank you, Professor Bertoletti. Now we will progress into the Q&A session. For the first question we have from the audience is, what is the difference between dendritic cell therapy and T-cell therapy? Which one is more specific and precise?
Well, okay, so what is the difference between a dendritic cell therapy and TCR-T cell therapy? So the dendritic cell therapy is designed to try to induce new T cells. So the dendritic cells are the professional antigen-presenting cells that are important for the induction of new T cells, both in, let's say, antiviral and tumor therapy. If the dendritic cell approach is working, meaning if the dendritic cells that have been, for example, loaded with tumor antigen will really be able to induce a tumor-specific T cell response, I will say the therapy could be extremely effective and very specific. Our therapy to basically produce directly the TCR-redirected T cells is basically instead try to engineer T cells that we know are already a specificity that can be able to basically target the tumor or the infected cells. Now, in chronic hepatitis B, for example, the problem of dendritic cells is that, again, if I want to induce new, for example, HBV-specific T cells, this is very complicated to do it in patients with chronic hepatitis B because the T cells are being already, the T cells specific for hepatitis B have been or already deleted or are exhausted. And in this case, the dendritic cell will not work. So again, it's a sort of different strategy. The dendritic cell approach can potentially work, I would say, if there are T cell precursor that can be expanded.
Okay, now we have the second question, which is between the EP versus retroviral T cells, how are you correcting for the cell dose in your comparison for effectiveness?
I mean, this is a very good question. I mean, the reality is that the retroviral T cells, and we know this already also from some data that we have in the first patient that was actually treated with T cells that were produced with retroviral, these T cells are growing in the patient. As such, in reality, the initial cell dose is just, I would say, a measurement of how many cells we are adoptively transferred. The fact is that the transient T cells, if they are working, they will expand in the patients, whereas the electroporated T cells will go down as a number because the TCR will be lost. So in reality, I think we cannot really make any sort of real comparison. It's just that the two products are working in such a complete different way because one cell is basically acting only for 2-3 days and then it's disappearing. When we are instead trying to do adoptive transfer with retroviral T cells, we are hoping that these T cells will expand in the patients. So I would say we have only now, it's time really to do more experiments, or actually not really experiments, but to try more in patients and see which could be the best effect. I just want to stress, however, that, we are trying now electroporated T cell mainly because we are still concerned about potential toxicity. And this is why, at the moment, we are doing this.
Okay, thank you, participants, for the questions. Due to time constraints, we are unable to answer all these questions. However, if you have more questions, you can email us at monkey.chong@merckgroup.com. The email address can be found in the announcement page. Before you go, please help us do a post-webinar survey, which will be sent via email to improve the webinar experience for the next iteration. Once again, I would like to thank Professor Bertoletti for sharing his research work and everyone in the Merck team who contributed to the success of this webinar. Wish you all a good day. Goodbye.
Goodbye.